Sure thing.
Welcome to Science Hangout from Science Mission. I'm Science Mission. Today we have Dr. David Hendrix, Assistant Professor of Biochemistry, Biophysics, and Computer Science from Oregon State University. Dr. Hendrix has published several articles in well-known journals. He's the reviewer of several reviewer for several journals and is a member of several organizations. Welcome, Dr. Hendrix.
Hi. Thank you for having me.
So let's move ahead and go through our questions. Can you please talk about yourself?
Sure. So as you said, I'm a professor of biochemistry and biophysics. I have a 2/3 appointment in biochemistry and biophysics and a 1/3 appointment in computer science. I do computational biology, and I'm interested in understanding how genes are regulated and using computers, algorithms, statistics, and probability to try and understand gene regulation in a broad sense, in a lot of different systems. So if you want to know a little bit about my background, I actually started off in physics and math. My undergraduate degrees are in physics and math, and it was a little difficult at first for me to go into biology because physics is all about universal principles. And then once you go into biology and you kind of find there is no universal principles, or there's almost an exception to every rule that you learn. But over time, with the advent of the high throughput sequencing and the genome revolution, I started to become more interested in biology because you're able to answer questions at a genome-wide level. You're able to study at a genome-wide level and compare different organisms, and biology is becoming more and more quantitative every day, and it is in some sense approaching something similar to physics in that we're learning maybe not universal principles, but at least common archetypes or motifs, regulatory archetypes that are used over and over again throughout many different biological systems. And so that's kind of where I am now, and certainly still interested in math and physics and always looking for ways of modeling whatever I'm studying. That's always difficult. I find genomic data is very difficult to model. It's in other words, as a physicist, you want to form simple models, and those don't always extend well to complex systems like a genome. So,
okay. You are busy being an assistant professor. Can you please talk about other extracurricular activities you do? Like, how is your off research? Do you do anything else? Your hobbies and other things.
I have a dog. I like to walk my dog. I do, the thing is, it's kind of funny for scientists. We like to spend a lot of our weekend times working on research, and it doesn't mean that don't have anything better to do. It just means that we're really interested in what we do, and it's exciting to go in and try and figure new things out. And then I think it's the whole thing that always drives me is the promise of new discovery. Once you've had a taste of discovery and that feeling of what it's like to see something for the first time, that's something that really has a high impact on myself and a lot of other people. But other than that, I like to go on hikes when I can, and I like to do drawings. I'm interested in drawing. I dabbled in drawing some comic strips, things like that. But lately, I've also been interested in drawing scientific figures, illustrations, that can help in papers, in manuscripts that I'm writing, but also educational material that I'm working on.
Oh, that's interesting.
I like to draw for fun too, and I'm a really big Star Wars fan. I don't have any Star Wars trinkets on my desk, but I'm a big Star Wars fan, and I spend a decent amount of time enjoying Star Wars and the Star Wars fan community and interacting with other Star Wars fans. So
that's good. And can you talk about your mentors, role models, and all?
I had a pretty unique background. I started off as a physics grad student, and my advisor was a physics professor who recently became a computational genomicist or genomics researcher. Genomicist, I guess, is what they're called. And at the time, in the early 21st century, there were no bioinformatics graduate degrees. There were no, in many cases, that whole notion of a professor of bioinformatics didn't exist. Nowadays, there are departments, there are programs, there are things like that. But back then, these were things being done by physicists and mathematicians that were starting to go into biology. And a lot's changed in the past 17 or so years. But my advisor was a condensed matter physicist, Dan Roxar, who got involved in a huge diversity of subjects throughout his research career, ranging from evolutionary biology to protein folding to genome assembly and annotation and to phylogenetics. But I learned a lot as a grad student, and I learned different ways of applying some of the math that's used in physics to studying genomes, I guess you could say. And then I did a postdoc in a Drosophila embryology lab for Mike Levine at Berkeley. And so my PhD was at Berkeley, and then I did a postdoc at Berkeley with Mike Levine. And so he's a famous Drosophila embryologist that has made a lot of important discoveries in terms of fundamental biology about gene regulation. And he's still making discoveries about fundamental biology of gene regulation. And I think when I was in his lab, that was what excited me, is even, we're basically at a time where the data was growing and growing and becoming more and more detailed, and you were able to answer questions by analyzing genome-wide data about gene regulation. So we found some new things about gene regulation and about microRNAs and how they're processed, and about something called shadow enhancers. We found these enhancers, these cis-regulatory elements or cis-regulatory modules, if you will, that are in some sense secondary to another enhancer. There's not a real obvious difference between a primary enhancer and a shadow enhancer, other than the shadow enhancers typically are evolving more quickly. They have lower levels of conservation, presumably because they're used less frequently. And maybe in some cases, they're more distal from the gene that they regulate. Anyway, we also found another class of small RNAs called MORs, microRNA offset RNAs. They're adjacent to microRNAs. And we found some other things about how RNA polymerase stalling might be regulated. So these are like really relatively basic questions about how genes are regulated. And then I did a postdoc for 3 years at MIT in Manolis Kellis's lab. And so in that time period, I did some work on small RNAs to see if small RNAs could be involved in regulating chromatin, but also some work in non-coding RNAs, long non-coding RNAs. And I was exposed to and began working on machine learning to a much larger degree. Some of the early stuff I did in DNA motif finding was machine learning in every in every sense of the word. But I think when I was at MIT at CSAIL, Computer Science and Artificial Intelligence Laboratory, I was exposed to a lot of powerful computational approaches from machine learning approaches like logistic regression, support vector machine, random forest, things like that. and just ways of evaluating them and ways of thinking about them, but also exposed to things like the importance of high resolution graphics, things that I've taken with me. You look at I always tell my students, you look at the high impact papers out there. One thing is they all have in common, not only are they doing really good, important science, but they present it in a way that's highly professional and highly organized and highly beautiful. In many cases, they're beautiful high resolution graphics to represent your data and to convey your point is really an important, valuable skill. And so those are some of the things that I picked up over the years. So.
Okay. And let's talk about your lab. Like, how big is the lab? How do you interact with your grad student postdocs? Just talk a little bit about them.
I am in my lab my office is actually in the middle of my lab. I have windows on my walls, and so students can see inside here. And they can see out. So there's pros and cons. The pros are I'm almost always available. The cons are I'm too available, to a point that it's hard to get away and do some writing on my own. So I need to sometimes either do that at night or find some other time to do it. But I'm highly interactive with my students, and I'm highly engaged with the work, almost to the point of micromanaging, although I try not to micromanage. But I think especially for students that are new to programming, it's important for me to look at their code. And so I do look at code. I do look at students' code, even when, especially when they're new to it. But after a certain point, I gain confidence, and then I can sort of focus on looking at the results rather than the actual code. And so, and then I just try and impart things on my students that have taken me 10 years to learn. I try and tell them at the beginning, so they don't take 10 years to learn those things.
No.
So.
Okay. So
but we work on a lot of different things. We're primarily a dry lab. We do computational biology. We're doing a little bit of experiments, actually. We're dabbling in some experiments, but it's mostly a computational lab working on genomics and some other stuff that's on the horizon that's not really genomics.
Okay.
But in the same vein, it's still kind of interesting. So.
Yeah. So how do you now correlate your genomics things to biology? You're not a biologist, so how comfortable and how have you progressed into the biology part of it?
I would actually call myself a biologist.
Wow. Good.
I feel like over time I have become a biologist, and I am interested in biological questions. My home department is biochemistry and biophysics. And part of the reason why that is because the questions that really motivate me and interest me are biological in nature.
Yeah.
And I want to learn new biological principles. I don't just want to find things that are already known biology. I want to find new biology. That's what really drives me. And I know a lot of biology. I mean, I've been doing, when I started off in DNA motif finding, it was a very abstract, almost mathematical way of describing something biological. It was just a sequence of A's, C's, G's, and T's, and I was looking for a pattern. And so in that case, it's very abstract. But later on, you find out that, those patterns are actually binding sites for proteins, for transcription factors. And then you learn, maybe these transcription factors interact with other transcription factors, and there's cooperative binding. So you're looking for pairs of motifs. And so over time, you build up a critical mass of knowledge, of biological knowledge, until eventually, here I am. I've been doing computational biology now for almost 17 years.
Wow.
So I think at some point I'm a biologist. I'm I'm interested in biological questions. I'm thinking about biological questions. I don't always know everything about biology by any stretch of the imagination. I'm always learning stuff. And it's important to always be a student. You know, I am still a student. I'm a student of biology, and I'm a student of machine learning. And there's things that I'm still learning, but you know, you just take it one day at a time. And so I don't know if that answers your question, but yes, absolutely, I'm interested in biology.
Yeah. Can you share your work with us, please?
Sure. So go to the talk.
Yeah.
So today I'm going to talk about, let's see, share this. And so we're good on the screen. Looks good.
Yep.
I don't mind turning this off, but either way, you want to just do this? Just try this. How's that look?
Yeah, it looks good,
okay,
as long as you can move around.
Yeah, I think I can. So basically, I'm going to talk about biological discovery through transcriptomics. And like I mentioned before, I think biological discovery is something that interests me. Let me just keep to my capitalization here. I probably should do that. Biological discovery and the idea that we can find new things, we can find new biology. This is what interests me. And so I'm going to talk about the technique of transcriptomics. And it's something that's kind of, in many ways, revolutionized biology in the past years. Okay, so simply, it's not, okay, let me just, yeah, I guess I do have to turn this on. Okay, so broadly speaking, this is a little schematic, a little illustration that I drew to shed some light on what transcriptome assembly is and what transcriptomics is. Essentially, you start off with a collection of isoforms in the genome that are transcribed into spliced transcripts, so they're spliced with the introns removed in eukaryotic systems. These are typically fragmented into RNA fragments, and then a cDNA library is built from these RNA fragments. And you may consider mapping them back to the genome. Some of the reads will have gaps in those reads in the alignment, essentially corresponding to the original introns. And the clusters of reads that overlap can correspond to exons. And so once the reads have mapped to the genome, in the case of reference-guided transcriptome assembly, you can build a graph representation, for example, where the nodes are the exons and the edges are the introns. And that can ultimately build into a transcript model, which ideally should recapitulate the original isoforms that you were trying to understand. And you know, this is what's known as deep sequencing. So this is essentially the only equation that I'm going to show. Well, I'll probably show a couple other ones, but just this is this notion of deep sequencing. You hear this expression. The depth of the sequencing is essentially the number of reads times the length of the reads on average, divided by, in this case, the size of the transcriptome, the total length of the transcriptome that we're trying to study. And so in essence, this describes the average number of reads that map to a particular exon in the genome, on average. And so one of the things that's really amazed me is the fact that now in 2017, or I guess this is from 2015, that the number of long non-coding RNAs actually outnumbers protein coding transcripts, or mRNAs. And in fact, to a large degree. If you look here, the link RNAs are actually 3 times the amount of protein coding genes. So when I was a grad student, you know, you pretty much think of these as the genes, but they're not. The genes are much broader set, including things that do not encode a protein. If you include these transcripts of unknown coding potential and pseudogenes and read-through transcription, it's essentially like 3/4 is non-coding. So I'm going to talk to you about 3 different stories. The first is looking at link RNAs, long intergenic non-coding RNAs that respond to abiotic stress in poplar. Abiotic stress, as we see, is cold, heat, drought. things like that, salt. And then I'm going to talk to you about age-onset rhythmic gene expression in Drosophila. And then if I have time, I'm going to talk to you about detecting cancer with stack denosing autoencoders. How much time do we have left? Like another half an hour or so? Or, okay. So yeah, we may not have time to get to everything, but I'll do my best. So first, to give an illustration of essentially this pipeline described here, this is essentially what we did. We took our FASTQ files, we quality filtered with Skewer, we aligned the genome with HiSat, formed BAM files with SAM tools, and essentially assembled a transcriptome with StringTie. And then what we can do is essentially we did a multi-step process to filter out things that had any signature of being protein coding or essentially not being non-coding. First, we filtered out transcripts less than 200. That's kind of a different filter. That's just the definition of long intergenic non-coding RNAs is that they're greater than 200 nucleotides. We filtered out anything with a significant coding potential from this program called CPAT, Coding Potential Assessment Tool. We ran other tools, Transdecoder, to filter out protein coding potential, blasted to Arabidopsis and essentially scanned for PFAM domains and landed on about 2,400 long non-coding RNAs. And then we basically filtered out those that had low expression. We call those significantly expressed. Essentially, their median is greater than or equal to 0.1 in all tissues. And that reduced it down to 930. Then finally, we said how many of these are significantly differentially expressed, meaning there's at least one tissue relative to control where these things are significantly differentially expressed. The result was about 100 exactly 100 link RNAs, with 14 of them partially overlapping transposons. So we kind of filtered those out as just transposon overlapping transcripts. Okay, so one of the things we may be interested in is looking at significant differential expression. So this is showing significantly differentially expressed link RNAs. So each column is a given link RNA that we identified. The rows are the different stress conditions that we evaluated and different tissues that we evaluated. And so you see we did 3 tissues, leaf, root, and xylem. And the stresses involved were cold, drought, heat, and salt, in addition to control. And then each of those stresses were for prolonged and short-term stress treatments. Okay, and then essentially, just to give a rough sketch, we found the majority of these long non-coding RNAs were being expressed under long-term salt stress for whatever reason in leaves. Long-term drought stress was also enriched for long non-coding RNAs in the root of these plants. And so just to reiterate, this is Populus trichocarpa, poplar. This is a common tree used in, for example, that they grow fast. They're interested, people are interested in poplar as a potential fuel source, for example, cellulosic ethanol, because you can grow a lot of biomass pretty rapidly. And so, you know, we're interested in how these things are regulated. And so one essentially characterized these long non-coding RNAs that respond to abiotic stress. And the other thing that we found was a lot of tissue-specific long non-coding RNA. If you want me to, I could probably do that. But basically, well, so here's one little bit of biology. This I is kind of like a weird thing. LNC RNA, long non-coding RNA, but sometimes you see an I there, and really that means intergenic, essentially meaning that it does not overlap a protein coding gene. But if you want to be more general, just this link RNA without the I is the most general class. So let's stick to that. So we were able to identify a number of tissue-specific link RNAs in our set. And so this is essentially showing the leaf, root, and xylem-specific link RNAs and their expression. And in this case, we're showing the expression relative to the minimum tissue. And we found 151 tissue-specific link RNAs. Okay, so one of the first things we wanted to do was essentially say, are these long non-coding RNAs? We don't really know what they're doing. We just know there's some transcript, right? It's not encoded protein. So first thing we can do is say, are there any significant correlations with other protein coding transcripts? In this case here, this is showing a particular long non-coding RNA. Forgive the names. They're all using these TCONs names, which are just automatically generated by the software. And this particular one happens to be significantly correlated with this protein coding transcript, hexosyltransferase. And so, you know, that's kind of interesting, I guess. But so then the other thing you could do is say, well, what if we looked at all mRNAs that are annotated to a specific pathway? And so that particular example, hexosyltransferase, happens to be part of this Xylan biosynthesis pathway here. So in this case, in this figure, we're showing for each pathway annotated in plant reactome, all the significant correlations between link RNAs and protein coding transcripts that are a member of that pathway. And so in doing so, we can associate link RNAs with pathways based on if they are correlated with more mRNAs than you would expect due to chance. And so we can build this kind of a heat map here, where what this means is you can zoom in here, like each of these little squares corresponds to a significant number of correlated protein coding transcripts. And so in this case, this particular TCONs, it happens to be correlated with GDP-L fucose biosynthesis. That's the pathway indicated here. And this particular link RNA here, this 14219 right here, happens to be significantly correlated with auxin signaling, which is one of these plant signaling pathways. It's sort of a plant hormone. Okay, so then when all is said and done, we basically have a pathway association for these long non-coding RNAs. We can't say precisely what the long non-coding RNA is doing. We can at least say there's a decent chance it'll be involved in that pathway. Okay, so the next thing we can do is say, are there any stress conditions that are associated with a pathway? And in this case, again, leaf salt being the most link RNAs, we see here, this particular dark band, it's linked to carotenoid biosynthesis and the TCA cycle, which maybe I'll get to a little bit later. These are kind of connected pathways in many senses, and there's a lot of genes that are sort of common amongst them. If you go back to this clustering, you can see this cluster here. All of these, you see carotenoid biosynthesis, Calvin cycle, auxin signaling, and TCA cycle is right here. But in many ways, these link RNAs are significantly correlated with a lot of genes linked to these pathways. Okay, so just as a couple of examples of some interesting ones, this particular link RNA right here, it's adjacent to this protein coding gene. It is significantly upregulated under prolonged salt condition relative to control. You can see control, very little expression. It's upregulated in salt. And it actually contains a protein coding transcript in its intron. So here's another one. Basically, this one was interesting because it was a really long intron, like 40 kb, 40 kilobases in length, but it's significantly downregulated in leaf drought. So it's expressed under control conditions in leaves, and it's essentially silenced under drought. And we have validated that this splice junction is really being used, and by designing splice junction specific primers. Okay, so anyway, the takeaway here is we can use transcriptomics to not only identify lots of new genes that are non-coding, but also potentially with enough different conditions, we can correlate them with protein coding genes and then ultimately associate them with pathways, biological pathways that they may be involved in. And we're essentially forming hypotheses as to what these things could be doing that hopefully can be carried out later on. So I next want to move on to a story in Drosophila melanogaster, which is, a common model system for studying biology because it is a simple organism that we know a lot about. So one of the things I'm interested in studying is the circadian clock, okay? So what is the circadian clock? It is a biological negative feedback loop, or a set of genes that are involved in a negative feedback loop where you have per period and timeless, they're forming these protein dimers that in turn repress clock and cycle heterodimers. And clock and cycle are sort of like the positive arm of the clock. They're transcription factors that will in turn activate lots of genes like period and timeless and like some other genes that also support the cycle. And these are post-translationally repressed by period and timeless. Light comes in, so light is obviously a big factor in circadian rhythms. We're exposed to light 12 hours out of the day. Light essentially activates this other protein called cryptochrome, which in turn, when light activated, will repress timeless. Okay, that's the circadian clock. Why is this important? So a lot of times people are more and more staying up, looking at their phones, looking at Facebook, exposed to more direct light. And not only that, aging is a big factor here. As people get older, they have less restful sleep, and they have sleep that is more punctuated by bouts of wakefulness. So what we wanted to do is we wanted to compare the expression of young flies to old flies. And so this figure here, clock, is that one transcription factor. And you can see it has this nice rhythmic pattern. After 24 hours, it decreases in expression, then comes back up. And there's no significant difference between young and old, okay? It's essentially the same profile. When we looked at period, to our surprise, remember period is the negative arm of the clock. It is actually increasing in amplitude at the mRNA level in old compared to young. So the expectation is with more period, we might expect more repression of the clock. Turns out that doesn't actually happen that way because the protein levels of period are actually decreased. I won't really talk about that, but it was interesting that previous papers had actually shown that period was decreasing with age. And the reason is because they were using this actin gene as their qPCR control, which is actually decreasing with age. So we found a new gene, DCP2, that's relatively constant expression in young and old. So confirming that period is in fact increasing at the mRNA level with age. So we found all these other weird examples that phase shift with age. Some of these genes actually have a complete shift, 12-hour shift in terms of the time that they're peaked as the flies get older. So we were able to generate figures like this that are heat maps. Each row of this heat map is a gene. Each column is the expression level at a particular time point. So you can see there's a clear rhythm going on here of high, low, high, low, high, low. But on the right-hand side, it looks like a low-resolution version of the left-hand side. It is kind of stochastic. These are genes that are rhythmic only in young. But to our surprise, we found a similar class of genes that are rhythmic only in old flies. They're stochastic or lowly expressed in young, but then they become rhythmic in old. So here's a snapshot of some of these genes that were validated by qPCR. And we actually continued this to day 75 old flies, which is kind of like the peak age of a fly, of a fruit fly. And then essentially a lot of these genes actually continue to increase in expression as the flies get older. We developed a two-dimensional representation we call the differential rhythmicity score that incorporates the degree to which the periodicity changes and the degree to which the amplitude changes. And so we can plot on a 2D axis here. Some of these genes like IMPL3, which is a lactate dehydrogenase, is kind of like the quintessential example. It's higher in amplitude, and more rhythmic. Other examples like HSP22 are significantly higher in amplitude in old, but not that much improved rhythmicity. It's actually HSP22 is slightly rhythmic in young flies, but it's just so lowly expressed that it doesn't really look rhythmic. So we were able to find these different classes, and we were able to identify what we call LLCs, late life cyclers, based on this statistical score. And we were able to confirm through qPCR that the LLCs increase with age. And then the other thing that we found is the LLCs are activated in young flies by oxidative stress. Oxidative stress, like reactive oxygen species, things like hydrogen peroxide, that are highly reactive and are one of the key stresses that we encounter during aging. Oxidative, we're exposed to more reactive oxygen as we get older. And so, therefore, these genes, for whatever reason, are activated by oxidative stress in young flies, and their expression in oxidatively stressed young flies essentially phenocopies the expression in old flies. Not only that, we next showed that clock, the positive arm of the circadian clock, is necessary for these genes to exhibit their LLC behavior. The exception being BNL here, which is actually better in wild type. The exception being, I guess, HSP22 here, which is actually increased in psych mutants. BNL looks like a true circadian gene because it's highest in wild type and then decreases to essentially low levels when you take out clock and psych. Some of these other genes are doing a little bit weird stuff, but some of them, as you can see, this HSP22 and maybe this one, are still rhythmic even in the absence of psych. So the other key result here is that these late life cyclers require light. They require exposure to light. If you put them in constant darkness, they go away. And that is, in some case, suggests it is not circadian. It is not strictly linked to the circadian clock, which does persist in constant darkness once you take away the lights. Okay, so one of the things that we found was going back to this idea of non-coding RNAs. We found this group of non-coding RNAs, novel non-coding RNAs, and who would have thought in 2017 that there would still be novel genes in Drosophila yet to be found? But we found these genes that are rhythmic in old and not rhythmic in young. And upon further evaluation, we have this one gene called crescendo that when we did qPCR, we were able to uniquely define qPCR primers. It's reasonably well conserved, and it's only expressed in old flies. That's why we were able to find it, because nobody looks in old flies. They only look in young flies for new genes. This gene keeps getting higher and higher with age until day 75. It sort of like looks exponentially more highly expressed with age, essentially. In other words, the day 75 expression trajectory dwarfs the expression in day 55. So when we looked to see what these non-coding RNAs could be doing, we found that they all essentially overlap annotations for what are called mature pyRNAs. PyRNAs are the largest class of endogenous small RNAs in animals. And pyRNAs are our genome's natural defense against transposons, for example. Transposons are these so-called jumping genes that copy themselves and reinsert themselves throughout the genome. PyRNAs are thought to combat transposon activity. They degrade transposons. So there is evidence now that transposons are actually active in aging fly brains. And it's not hard to imagine why they would be really harmful for for an aging system. If you're inserting transposons over the place, it's going to wreak all sorts of havoc, disrupting protein coding genes, disrupting regulatory DNA. So we believe these pyranase are becoming active only in old flies, specifically to combat age onset stresses due to transposon activity. So that basically concludes my section on the LLCs, late life cyclers. And the takeaway message there is that the late life cyclers, to a large degree, we think they could be part of a stress response pathway that's activated late in life. So how are we doing on time?
Yeah, you still have about 8-10 minutes.
Okay, I could briefly touch on this deep learning stuff. And everything that I've talked about so far is kind of looking at statistically significant genes in terms of what's the most highly expressed. But we wanted to know, can we move away from that? Can we use machine learning to move away from that? And one of the questions that we had was, can we detect cancer? Can you tell that a given cell is cancerous versus non-cancerous based on its gene expression? Okay. And we wanted to use deep learning. The reason why we want to use deep learning versus ordinary machine learning is there's more layers, which kind of gives more detail. You don't have to do feature engineering. In these layers, it sort of encodes its own features, meaning it learns what's important in the data rather than you telling it what specific quantities it should be used in the machine learning. So it's capable of learning features and it's capable of learning nonlinearity in the data. So this is a picture of Google's Deep Dream. It's kind of unsettling. This is basically what they did is they took a convolutional neural network, trained it to recognize dogs, and then they basically mutated the image, taking mutations that improve the classification for being a dog. And it produces these weird effects that it's important to remember. With deep learning, we want to be careful with what we're doing because sometimes the computer can go a little haywire and think this is a dog. No person would say that's a dog. So we want to be careful with how we interpret the results from deep learning. We used what's called stacked denoising autoencoders. So one of the challenges for studying gene expression, especially cancer, is you're dealing with this dimensionality curse here. You have a high number of features, which are your genes, 20,000, 40,000 genes, and a limited number of samples, typically on the order of 1,000 in this day and age. And so we need to either get more data, get more patients to give samples, or what we can do is what we did, and that is to do dimensionality reduction and to reduce the dimension of the gene expression data so that it's on par with the number of samples that we have. And so this is what's called an autoencoder. It's a basic dimensionality reduction. It basically encodes the data to a reduced dimensional representation in such a way that it can reconstruct the original data that's true to the original data. It's accurately reconstructing the original data from this reduced dimensional representation. And mathematically, it's basically a linear or affine transformation with the data times a matrix plus a vector, and then applying this nonlinear sigmoid function or nonlinear squashing function. And you can do, in addition to just doing one layer of an autoencoder, you can do a stacked autoencoder where you actually iteratively reduce the dimensionality of the data. And by doing it iteratively in this sort of stepwise fashion, you lose less information than if you did it all in one jump. Then we can do other strategies to avoid overfitting. One of them is called denoising, where we're actually essentially adding noise to the data here, stochastically modifying our data. And then also dropout, where we stochastically remove various nodes in our layers to sort of train more robustness into the model. And so when you put it all together, you get a stacked denoising autoencoder. And so we took the gene expression data from cancer patients and healthy patients, reduced the dimensionality into this new feature space in such a way that we could reconstruct the original data. And then we can use this new feature space to do machine learning. And basically, in this case, we're looking at ovarian and prostate cancer. And I'll skip over that. But we compared it to some of these other machine learning approaches, single layer artificial neural networks and restricted Boltzmann machines and deep belief network were some other approaches that we compared our model to. So in the case of classification, what we're essentially doing is we're learning this reduced dimensional representation, and then we're taking that data and feeding it into these traditional machine learning approaches like the single layer artificial neural network, Boltzmann machine, and deep belief network. And our accuracy, we get about 92% accuracy here for the ovarian cancer using the neural network, and about 91.29% accuracy for prostate cancer when we use this traditional single layer neural network on our reduced dimensional data. So had we done this on the unreduced data, it would be worse. And we'd be dealing with that dimensionality curse. We wouldn't have enough data to train the parameters that we need. So we had this idea, we have this iterative mapping here, these matrices that are reducing here. What if we take the product of all of our matrices? And so this is in some sense linearizing a nonlinear transformation. But what happened was when we did this, we got this like nice Gaussian fit of these products. So we take the product of all these matrices and then ask what genes have the highest weights in this product matrix. By fitting this to a Gaussian, find the statistically significant weights up here, and we selected a subset of genes that had statistically significant contribution to the dimensionality reduction of the gene expression data. And when we trained on this new data, these highly interactive genes, we actually got a much smoother convergence during machine learning with the restricted Boltzmann machine than we did with the full set of stacked denoising autoencoder features. And when we applied our machine learning approaches to these deeply connected genes, in the case of ovarian cancer, we actually got better accuracy. So we essentially used machine learning to identify a subset of genes that are not necessarily the most highly expressed. So typical approaches will just say what's highly differentially expressed. We're able to find the most meaningful genes based on the degree to which they contribute to a dimensionality reduction of the cancer gene expression. We get similar results for the prostate cancer. And when all is said and done, we find a lot of genes that are enriched in that set. And a lot of these genes, as you might expect, are related to Go terms that might be associated with cancer, things like cell death, apoptosis. So we're able to find these genes that seem to be relevant and something that we're going to pursue a little further and figure out what these genes are doing. And I would like to acknowledge the students and other people that did the work in the Poplar project. This was a collaboration with the JSWAL lab here at Oregon State, supported by funding from the Department of Energy. And that was the work of my student, Michelle Wiley, and the Circadian Rhythms and Aging project was the work of a former student, Rachel Kinsel, in collaboration with the Gaboltovich lab. And Eileen is a research scientist on that team. And then finally, for the stack denoising autoencoder, that was done by Madisa Danier and Reza Ghani. And so with that, I will conclude the presentation. And thank you.
Yeah. Can you unshare the presentation, please?
Sure. Let's see. How about that? Nope.
Let me see. Yeah, that's good. Yeah. I have just a couple of, because of the time thing, I have a couple of questions. Sure. One is regarding the long coding RNAs.
Sure.
So there is disproportionate amount of RNA compared to these RNAs compared to the proteins and all.
Right.
So we know that there is a lot of regulation at the non-coding region happening and all. So like so many RNAs play roles somehow in regulating so many genes, or is it just like some are redundant? How do you those kind of RNAs out there?
I think it's still a work in progress. I think we're still learning what they do. I mean, if you take a look at small RNAs like microRNAs, a given protein coding transcript can be regulated by dozens of distinct microRNAs. So it's possible that a similarly large number of long non-coding RNAs can be involved in regulating a given gene. But at the same time, link RNAs do other things other than just regulating protein coding genes. They play structural roles in molecular complexes. I mean, a good example is telomerase. There's an RNA called TERK, the telomerase RNA component. And it is an interesting long non-coding RNA. And there are actually disease-associated allelic variants in that transcript. So in other words, there are mutations, genetic mutations that are associated with diseases, and they're not in a protein coding gene. They're only in that long non-coding RNA. Presumably, mutations that affected structure, for example, possibly. So, not to mention the classical non-coding RNAs like the ribosomal RNAs. These are things that we've known about for decades, and they play important structural roles in the ribosome. So I think really we've only begun to understand what long non-coding RNAs are doing. They're scaffolds for molecular complexes. They're regulating genes. They're recruiting genes. I think there's a good chance that they operate through sequence-specific patterns, sequence-specific activity, much like microRNAs do. And maybe, who knows what they do? Maybe we'll have something. Maybe it turns out a lot of them are doing something we haven't even conceived of yet, for all we know. So.
Okay, and the last one is
by the way,
basically.
That's what makes it. Sorry to interrupt, but
interesting.
You had asked.
Sure. It's okay.
Early on, why non-coding RNAs? But that's what makes them exciting. And I think a lot of what I've talked about is genes, long non-coding RNAs, and why they're regulated. But I think the really interesting question is how they regulate. What are the molecular mechanisms these things are using to regulate other genes and to do what they do? And I think that's only understood for just a dozen long non-coding RNAs, maybe, if that. So.
Okay, and lastly, the genes which through the machine learning, like you have the cell death pathway and all those things. There are a bunch of genes are associated with the this pathway or like cell death mechanism. So how do you putting them together? Like, I mean, like there is a known pathway, a few known pathways in different organisms, different cell things. Now you are talking about another 100 of them. You are bunching them together. So do they play some role like outside, like minor role outside, or like they do have a major role on specific things, specific cell death pathway? What is your, like, I mean, like it is still too early again, as you said, for non-coding RNA.
I think it's...
Think about currently.
I think the takeaway there is that maybe we shouldn't just, a lot of previous studies have just said, well, what's the most highly expressed in cancer? Well, maybe that's not always the most meaningful. Maybe there are genes that are more subtly expressed, but are still playing more critical regulatory roles. And the hope is that maybe by doing some of this machine learning, we can identify those more subtle examples. Okay. What would we do with them? In the short term, perhaps they could be used as biomarkers in a cancer diagnosis. If you have a tissue sample and you could just, if you knew those genes, you could simply test the expression of those genes. With that said, that's also tricky because if they're more subtly expressed, it's harder to know, it's harder to use them as biomarkers because maybe their change in expression is not that drastic. But the hope is that maybe this will enable us to focus our attention on these new genes, or these genes. They're not new genes, and in some cases they're not necessarily newly implicated in cancer, but maybe this will serve as a way to change our focus and to look at studying what these genes could be doing. But it's clear that they're relevant to cancer expression, cancer gene expression.
So I think with that, I think we'll conclude this Hangout. Thank you so much for taking time. It's pretty interesting. In fact, I learned a lot from whatever you have presented. This is like I haven't worked so much on all this stuff, but certainly it is a very new field, even though it's been done for a little bit, several years. But I think things are opening up right now, and I think we hope to see more of your work, more of your publications in the near future. Thank you so much.
Okay, thank you for inviting me.